So let me firstly introduce Steve Griffin, who is a scientist at Leeds University. Steve's purpose for today is to talk us through the background in relation to vaccinations, virology, and just to inform us with a bit more information so that we can support our colleagues and employees and also provide them with that direct information as well. So Steve's got a number of things that you'll be running through, haven't you, in terms of topic points, which I've also shared in the agenda. But please, there will be at the end an open Q&A session. Feel free to ask questions. And if you or if anyone that you're aware of would like to have a follow-up one-to-one session, sort of a short call with Steve, we can arrange that. Just let me know direct if that's okay. So without further ado, I will mute myself and hand over to yourself, Steve, if that's okay.
Yeah, sure. Thanks ever so much, Claire. And thank you everyone for making time to come and listen to this. So this is just a really, really informal opportunity for you guys to ask any questions that you might have or concerns that you might have around the vaccine. I should say straight from the start that I have no ties whatsoever to any companies or laboratories that are involved in making vaccines. I'm a virologist. I do work with the coronavirus in a high containment level laboratory sometimes, although I usually get younger people than me to do it most of the time nowadays. So I do have a sort of first-hand experience with this virus. And I hope to persuade you today that the vaccine is definitely the solution to our current predicament, regardless of what may have been said yesterday. So I will absolutely try my best not to get drawn into politics unless anyone has any specific questions about policy. That's fine, but politics I'm not really going to go into, if that's all right. So what I'll do, if it's all right with you lot, is to just take you through a very brief introduction around some real fundamentals of biology and a little bit about viruses and our immune systems and why vaccines are so useful and so important in terms of how we manage diseases in the present day. What I'm hoping to do is to give you analogies to try and cut through a lot of the acronyms and jargons that you may hear, which are floating around the place. And it's the sort of way I conceptualize things as well. So please, I hope that you don't feel like I'm patronizing or anything like that. If there's any questions, please feel free to just unmute and interject and give me a shout, really. If I start rambling as well, do that as well, please, Claire, because that will happen, okay? So I'll do that. I'll try and explain then why it's important for us to have our vaccines and why they're very safe and very, very effective. And then I've got a list of sort of common concerns that people have, which I'll try and address. I have a strong feeling against some of these things that people post on social media, which are inherently untrue, and we'll try and go through some of those common things today and discuss the actual science and data and understanding behind some of those issues. I would like to say from the outset, though, please don't feel as though there's anything wrong with having concerns about this vaccine. This is a stressful time. We've had a torrid time in the last couple of years. It's a difficult situation. Everyone's worried. Everyone's, fed up with all the restrictions and the endless cycles of lockdown. So, we really need to ensure that these vaccines are used to their best possible means to get us out of this. Okay. So please, as I say, please feel free to ask questions. Has anyone got any questions at the start that they'd like to ask before we get going? No? Okay. So the first thing I want to talk to you about is really how our bodies are made up in terms of how our bodies are organized, and they're organized into cells. And these cells are tiny little building bricks of our bodies, and they're controlled by the genetic information that's in our genome. And our genome is made of this stuff called DNA, which is literally very long strands of endless repeats of these 4 little letters in different combinations. And those 4 little letters are actually powerful enough in terms of the number of combinations they can do to create us. And frankly, going from a single cell to us is a staggering piece of biology, which I don't pretend to understand nowadays, because we now understand far more about it. But we need to understand some fundamentals about how those cells work in order to understand what a virus is and what a virus does, and then also to understand how our immune response is geared up to try and stop that happening. Okay, so an analogy that I tend to use quite a bit is, if you think about these cells, these individual cells in our body, which can be anything. They can be in our eyes, in our earlobes, or in our left arm, muscles, heart, nerves, highly specialized things, but essentially they boil down to a storage point of information and a means of getting that information to be made into something that does a job. Okay, an analogy I actually use is a restaurant, which is a bit of a strange analogy, I realize, but if you can conceptualize our genetic information stored in the nucleus, which is a small segregated part of that cell, the DNA. If you think about the nucleus and the DNA there as 46 massive cookbooks, okay, huge lists of recipes, which give us all the information about all the possible dishes that you'd ever want to make in your restaurant, okay? That's a massive lexicon of food knowledge, and that's us, really. An actual fact, not a huge amount of it is actually used to write down recipes, but that's another topic of discussion. And then what obviously you need to do to make a dish in that restaurant is to get a recipe. And
can you hear this chap speaking? Can you hear this chap speaking?
Yeah, we can hear you too, Chris. Sorry.
Do you have a question, Chris?
Oh, sorry.
Do you have a question? No?
He's gone. Sorry.
Okay. So, yeah. So I apologize for this.
Struggling to hear you at the moment.
Oh, sorry. Can anyone else not hear?
No, you're fine.
Yeah, you're fine.
Okay. Well, sorry about that, Chris. This is recorded, so I do believe you'll be able to watch it back if you're so inclined. I don't know. Right. So where was I? Yeah. So you want to make a meal in your restaurant, okay? And that meal is what we would call a protein. So something that does a job. And to get that meal, that protein, you want the recipe, and that recipe is what we call a messenger RNA, okay? And the job of that RNA is to take the information for that dish from your big lexicon of cookbooks out into the kitchen, and the kitchen is what we would call a ribosome in the cell. So that's the protein making factory. And we encode something like 20,000 different proteins in every cell of our body. And the different combinations of those proteins, the way they interact with one another, is how a cell comes into being. And it has a lipid membrane, but everything is organized by the proteins that our genome encodes, okay? So if you just remember this, it's something called the central dogma of biology. DNA makes RNA, makes protein, okay? There are some awkward processes, things like retroviruses, so things like HIV, which go the other way, but we're not going to talk about those today, okay? Although we might do, actually, addressing some of the concerns about vaccines later. So our genome is something like 6.4 billion letters, all right? That's a lot of information, and it's all just made up of these 4 little bases in different combinations, and every 3 bases code for a chunk of a protein called an amino acid. That's how our genome works. It's literally just strings of code. It's like something that a computer might read, but it's in a base 3 and base 4 coding capacity. Okay, so this is our restaurant, and usually our restaurants are very defined. There are different types of restaurants doing different jobs. Okay, those are the cells in our body. Now, what's a virus? Well, a virus really is like a small pamphlet of recipes. It's a very small piece of genetic information, okay? So to put it in context, there's our 6.4 billion letters of code in our genome, and the SARS coronavirus 2, the thing that's causing us all the troubles at the moment, is just 35,000, okay? Now that still sounds like quite a lot, but actually it's 200,000 times smaller than our genome, okay? It encodes for a handful of proteins, something like 20, okay? So 1,000th of the protein coding capacity of our bodies. And yet this remarkable sort of sub-recipe, sub-cookbook is able to get into our cells and completely circumvent our body. And for me, that's part of the reason why I study virology. I find the fact that something that is so limited and primitive by comparison to us is able to completely circumvent us, okay? So an actual virus itself is small. It's something like 100 to 200 nanometers, which is 10 to the minus 9 of a meter a nanometer, which is, oh goodness, a millionth of a millimeter, okay? So it's small, right? And all it is this tiny piece of genetic information. In the case of this virus, it's an RNA, so it's already ready to be read by those factories that make the meals, okay? The ribosomes. It can make what it wants to make, its proteins, immediately when it gets into the cell, but it's tiny. And it's there, and it's coated in a protective protein coat, which we call a capsid. And then this particular virus also has a lipid membrane around it, much the same way as our cells do. And on the outside of that membrane are tiny little proteins that stick out. You may have heard this because it's the focus of most of the vaccines that we're going to talk about. The spike protein, it has some others, but the spike protein is what we're really talking about here. And it's that protein that hooks on to our cells and acts as the sort of key and lock mechanism to get them inside of our cells. And once that virus is inside of us, it starts making its own proteins, and through a very complicated series of interactions, it basically hijacks us. It takes the cell over, it corrupts the cell, and it turns it into its own sort of restaurant, okay? SARS likes its own sort of food, and it will make the cell behave accordingly. And the reason it wants to do that is because the sole purpose of this virus, this little piece of genetic information, is to copy itself, amplify itself, and spread on to somebody else that's susceptible. So transmission is the absolute raison d'etre of a virus. That's all it ever wants to do. It's just an unfortunate side effect of that process that it makes us really quite unwell from time to time, okay? So a virus is what we would term an obligate parasite. It absolutely needs us to thrive. It's the ultimate example of a selfish gene, but it hasn't had the wherewithal to go and create a body around it. It's literally a piece of information that passes from one of us to another and then hijacks us in order to make copies of itself and pass on, okay? So if you think about that, keep that in mind, the transmission is really what this virus wants to do. And so we've obviously experienced this through history, throughout our entire evolutionary history. And if you remember, I mentioned that a lot of our genome isn't actually used. Well, actually, a good proportion of it is actually virus sequences that have become part of our genetic information over the millennia, okay? But I won't dwell on that now. It's a different process. But we have been living with viruses for eons, okay? And part of the thing that we have done to try and live with viruses is to evolve this very complicated, but actually relatively simple in principle, immune system, okay? So the immune system is our defense. It's designed to recognize things that are not of our body and to target a response accordingly, okay? There are different layers to this immune response, okay? Some of it is like laying a trap and, you know, depth charges and tripwires and things which will trip over an enemy invading pathogen, okay? And there are sentinels that spot these things in our cells and can recognize them. But the really important aspect of our immune response is what we call the adaptive response. And the adaptive response is so called because it can exquisitely target these invading pathogens and mount a response that's entirely controlled and appropriate to destroy that incoming invader, okay? Now, much is made of our immune response. You'll see it on adverts on buses, things like, take this and your immune response will be massively healthy, and if you eat this, that, and the other, then you'll have a fantastic immune response. Now, there's an element of truth to that because our immune systems are sort of trained by things like our natural environment, the bacteria that live in our gut, for example, and having the right nutrients is always very helpful. But actually, as long as you have a relatively normal balanced diet, and you're a relatively healthy person, there's no reason why your immune response, this adaptive response, won't work in any way, shape, or form. It does get weaker as we grow older. It's literally, it just runs out of steam a little bit. And there are various conditions that some people can have, or some medicines that some people can have, that can dampen the ability of that immune response to work. Okay, and these are some of the people that we're really worried about when it comes to infectious diseases. So the idea that our immune systems can be trained by any of this sort of, slightly quackery ideas of weird diets or taking huge amounts of vitamins, which our bodies regulate to very specific levels anyway, I'm afraid that is a bit of a misconception. Obviously, you don't want to be deficient in vitamins and things like that, or nutrients, and you don't want to have any conditions that affect your immune system. But realistically, the vast majority of us have a perfectly well-functioned immune system. So our immune system really isn't like a muscle or something that you can build up and be stronger. It doesn't matter if you're Arnold Schwarzenegger or Adrian Mole. These things, they will work. Okay, your bone marrow is churning out these cells which are exquisitely programmed to do a certain job. So I prefer to think of it actually as, rather than a muscle, as actually a sort of an arms locker. And I say that because your immune system is actually incredibly powerful. Okay, it's extremely clever, it's adaptive, but it's also very powerful. And you can see that in certain diseases where it goes wrong. So people that have autoimmune conditions, for example, where their own immune system is attacking parts of their body because of slight mismatches in their genetics. And that is an important thing to remember. We are all genetically diverse. We all have things that are going to be working ever so slightly different between us. But as a population, by and large, our immune response is intact. Okay. And I refer to it as an arms locker because literally what you want to do is open it up when the time is right, take the correct weapon out to combat the pathogen that you're dealing with at that time. And the most important thing is that when that battle is won, you need to put it back and put it away, probably in a place that's very nice and accessible for the next time you might see something, but you need it locked away again. Because if our immune systems go crazy, which can happen and is actually part of the pathology associated with COVID, then that can be really bad for our bodies. And any infectious process that we experience is a balance between the inherent damage caused by that invading pathogen and our responses to it. So it's a little bit like the sort of collateral damage you would see on a battlefield. Okay. So the reason our adaptive response is so incredible is that during our early embryonic development, it amasses this huge array of white blood cells with a little sensing protein on the surface of the cells, which can basically recognize masses of combinations of different chunks of proteins. Okay, not the proteins from our own bodies. Those cells are deleted, so we don't recognize ourselves as a foreign invader. This is quite important because otherwise we wouldn't make it much past an embryo. So we have this trained immune response, this weapons locker, which can recognize all variety of patterns and chunks of proteins that come from outside of our bodies. And so what happens is when an invading pathogen comes into our bodies, our cells that are infected by that pathogen, they try and nibble bits of that pathogen up and show it on their cell surfaces. And there are special cells, special white blood cells in our bodies that do this incredibly efficiently. And when those are presenting this part of the pathogen to our white blood cells, that's when the arms locker gets opened. That's when our immune system is triggered to do the specific job that it absolutely must do. Okay. And there are two branches of this response. Three really. One is these sort of traps and mines that I mentioned before. That's what we call our innate immune system. And that actually, if that's triggered, that helps the more specialized forms of things kick off. But then there are two sets of soldiers that we employ, okay? And they're all targeting this foreign entity. They're not targeting our own bodies, okay? So the first branch of this response is what we call a T cell, okay? And the T cell's job is to look around to see cells that are actually hiding a virus inside of them, Okay, so they'll be presenting these little flags, parts of the virus, and saying, look what's inside of me. And the T cells' job is to come along and literally kill them. All right, so a massive part of our immune response is actually kill the cells that have a virus in there. So that's part of the reason actually why we get slightly unwell. And a lot of the chemical signals that are involved in that process are what give us the symptoms of a cold, and they kick off fevers and things like that. So that's a major aspect of it. So that's recognizing the virus while it's sort of doing its job of its recipe. Okay, so it's inside of our cells, subverting those factories to making more virus particles. The second branch of our immune systems is almost like the artillery. Okay, these are called B cells. And what B cells do is they make antibodies. And antibodies are small proteins, which again, exquisitely recognize, they're trained to recognize these small fragments of proteins that are invading, they're non-self. And what they're recognizing actually though, is the proteins in that virus particle itself. So the antibody's job is to stick to that virus particle to prevent it getting into your cells in the first place. So you've got two layers there. One is the sort of protective sort of flak layer, stopping things coming in. And that's part of the thing that we want to do with vaccines, is to train that response to happen. And I'll come on to that in a moment. Okay. So your antibodies will stick to a virus, stop it coming in. And if you are unfortunate enough to be infected by that virus, then your T cells will go along and literally execute any cells that have that virus inside of them. Shall I just pause there and ask if anyone has any questions? Does that make sense? Am I rambling? Okay, right. So what happens in a normal infection is that you'll see a virus for the first time, and this is also true for bacteria and fungi and all the rest of it that invade our bodies, even sort of parasites and things. They have different approaches to dealing with it in our immune response, but a virus is the one we're talking about today. So when your immune system first sees that virus, it's got a job to do, because it's got to mobilize all of these aspects of its immune response. It's got to get to the weapons locker. It's got to unlock it. It's got to pick the right one. It's got to get things going. Okay. So often we're behind the first time that we see a virus. And this is part of the reason why SARS coronavirus 2, the virus that causes COVID-19, is so devastating, because it's jumped across from an animal host and we've never seen a virus like it before. Okay. So this is why we have no response to it at first. And that's why a lot of people get very sick, because they can't deal with this. Remember, we've got this vast array, this repertoire of things, and for various reasons, partly because the virus is quite good at avoiding our immune system, which I won't talk about today because that really does get complicated. But this really is a problem. And that's part of the reason why we've had to put in such horrible restrictions throughout this pandemic, because before we've had vaccines, we've not been able to do anything about it. So normally what happens when you first see a virus is you make this response, and it's a little bit slow, it's a little bit weak, but it's just about enough eventually to catch up with that virus and beat it. Sometimes it doesn't happen. Sometimes it all goes a bit wrong. Sometimes people will succumb. Most of the time, thankfully, people will fight it off. And we see this throughout our lives with different viruses that we have. Some of this make us sick for a very long time. Some of them don't. But what the trick is that the body plays is that it uses this experience to make sure it doesn't happen again. And what it does is certain white blood cells that have been amassed to fight that virus, some of them, most of them actually, they commit suicide when the virus is gone. They're programmed to commit suicide. They're all sort of identical clones of each other, but some of them stick around, and those are called memory cells. And those memory cells are standing ready in our body. So if we see that pathogen again, it's much easier to unlock that arms cabinet and get things going. Okay, and our memory response is an incredibly vigorous response and incredibly protective. And we also keep antibodies in our bloodstream throughout lives at various levels that can be re-stimulated again, which again even stops those viruses getting into our cells in the first place. So that's what we term immune. Okay, it's a relative scale. It's not always 100%. and things can happen that change that. But that's what we call immunity, because the second response to that pathogen is so strong that it prevents the infection from happening again. So as I say, the natural, that's probably the normal process by which this happens is for us to see this virus again and to have had an infection early on in our lives or now or later in life that primes that immunity. What vaccines do is they trigger that memory response, that second response, the strong response, the protective response, without the slight inconvenience of experiencing a potential lethal virus infection. And so that is the whole concept of a vaccine. It's basically to spare us from having to go through that first infection to get our weapons cabinet open and ready for the next time we see it. And the way that the vaccines do this, they've become more sophisticated in time. If you remember the original story with Edward Jenner, who took some pustules from a milkmaid and scratched it into a poor young lad's arm and then exposed him to smallpox, I think we'd struggle to get ethics to do an experiment like that nowadays, okay? But what our vaccines are doing now is that they are specifically making one recipe, one dish from that virus, mainly the spike protein, which is the one on the outside of the virus particle and hooks into the cells. And what that does is elicit those armies of responses, the T cells and the B cells, to get ready and make a memory of that response so that when we see SARS coronavirus 2, hopefully after we've had our vaccination, the response is so strong that we are immune. Now, again, we're genetically diverse. There's a sliding scale of that. There are gray areas. Sometimes we might get infected, but the infection is very, very mild and we don't pass it on. We have changes in the virus, which we can talk about later, if you like, in terms of variants, which may mean that our immune response from the vaccine isn't quite as good. But by and large, the vast majority of people, unless they have a problem with their immune system, make excellent responses to these very sophisticated vaccines, and they literally spare us from getting ill and protecting us in that way. So does anyone have any questions about that concept, about the idea that a vaccine is training our second response? No? Okay. So now on to the vaccines, and I'll start to now touch on some of the sort of common concerns that people may or may not have about these things. Now, we'll have heard all these terms bandied around by people: RNA vaccines, adenovirus vectored vaccines, so like the AstraZeneca vaccine. The RNA vaccine is things like Pfizer and Moderna, okay? All those are doing is cutting to the stage of transporting that recipe into those cells that are very, very good at presenting the information to our white blood cells, okay? So for an RNA vaccine, it's formulated, it's literally one of those recipes for the spike. It is coated in a lipid formulation, which allows it to pass into our cells. That lipid formulation then dissipates. And that RNA, that recipe, goes into the factories, into the kitchens in those cells. It's made, it's presented in that cell just in the same way as if it was infected with a virus. And it flags up those professional cells to come and say, oh dear, this cell's potentially infected. So let's make our T cells, let's make our B cells, let's make our antibodies and train our soldiers so that next time we see this, that's what's going on. So you do see a reaction, a bit of pain, and sometimes, more pronounced fevers and things like that to vaccines, okay? Because you are actually making an immune response to them. So that's why we feel a bit rubbish sometimes after our vaccines. There are some more extreme versions of that, which we can talk about in a bit. The virus vectors do exactly the same thing. And I should say that the RNA vaccines, they're not stable. They don't hang around. RNA itself is inherently unstable, because otherwise you'd be making the same recipes over and over again in your cells, and that would not be a good thing. So these RNAs are slightly stabilized, because otherwise it wouldn't work very well, but otherwise they just are degraded by the cell's natural mechanisms. And the viral vectors are exactly the same thing, but they're actually DNA. And so they're just a hard copy of those recipes. and they cannot replicate themselves. They've had parts of their genome removed, but they have this recipe from the SARS coronavirus 2 in them. And it's like having a little sort of mini nucleus in your cell. It goes in, makes the recipe, again, just that single protein, and trains your immune responses. Okay. Now, one of the main concerns that people have over these vaccines are that they are relatively new technologies, and that they were supposedly rushed through to being approved for use in humans. Okay, so I'd just like to pick up on those as a starter. And now, really, please, everybody, just chip in and ask questions if you want to, because I'm past the tutorial now. So I apologize if that was a bit dull, but it really is important that we understand those fundamentals if we're going to go on and talk about the actual science behind these vaccines. Okay, so these vaccines are certainly new in the sense that we've never had them rolled out in humans on such a scale. Okay. But there have been trials of both of these forms of vaccines in people many times before. But the problem with vaccine development is that it's basically demand driven. Okay, so the Oxford labs and Pfizer and Moderna, they were all doing small scale studies. They've all passed safety tests in animals, and they were all doing small scale studies for other diseases. Some of them are related to COVID, to SARS coronavirus 2. So like the MERS coronavirus, for example, was one trial that was being done. And we know, therefore, that these technologies really do work extremely well, and they're very safe by and large. Okay. So when the call came back in 2020, my goodness, this is a pandemic, alarm bells ringing. We need to make vaccines as quickly as possible. The actual technology itself is pretty straightforward to do. I mean, I could ask one of my PhD students to make the equivalent of the AZ vaccine in less than a week. We work with those viruses, and we can do that. That's absolutely fine. And nowadays it's very straightforward to do that. The trick is to make them in a sense that is to a completely correct clinical grade, and to ensure that the coding that you use to program the immune response is actually going to be the right thing to use. So those are the real difficulties there, but the technology was already there. Okay, so they managed to go into mass production and make these vaccines. Remember, they're non-replicating, they're not live vaccines. These are artificial ways of expressing that coronavirus protein. So they were able to rapidly ramp up that procedure to make lots and lots of vaccines, and that enabled them to go into trials. Now, it's very true that the trials were done quickly. That does not mean that they weren't done properly. And there are three reasons for this, okay? Usually the things that hold a vaccine trial up are, one, red tape. And believe me, I've done clinical trial applications. It takes forever. So one of the biggest exercises that was done was lots of collaborative and discussive work with all the regulators, enabling people to smooth that process through and not have to wait for weeks and weeks and weeks for someone to even pick a piece of paper up, which is usually what would happen. Okay. So that's usually a massive bottleneck in getting any kind of trial through. And remember, you have to go from one, which is safety in healthy people, to two small scale studies, to three are really big trials that are geared up to look at the efficacy of those vaccines. Okay. And in fact, there's a fourth. I've forgotten the fourth. So the second aspect of that is, rather than going through the first, second, third phases, they were able to design those trials so that they actually rolled on into one another once certain checks were made. So rather than having to go through, publish, get it reviewed, they just went straight the way through as soon as they understood that it was safe. Okay. So they were able to overlap aspects of the trials to accelerate the progress there. Okay. The next bottleneck is money. Usually, to get money to fund a trial is difficult. You need to get a pharmaceutical company involved at later stages. And of course, you don't always generate the interest for that. And so the investment is usually a huge limiting factor in any study that we try to do in vaccines. But of course, the governments of the world were all funding the hell out of all of these things, as were the pharmaceutical companies. And remember, AZ does this at cost. So, I think that's incredibly altruistic of them to do that. So cost was removed as another barrier to developing these vaccines quickly. And the last thing that's really important in accelerating a trial through, which is actually something that people just, it's there in front of you, but you don't often realize it, is one of the biggest restrictions that you have in developing vaccines is the number of patients that you have available to test it on. Okay. Usually recruiting for clinical trials can take weeks and months to even recruit a handful of patients. But fortunately, with a pandemic on, there were quite a few about. Okay. So what that enabled us to do was, because the virus was present in such high numbers, is to randomly give people the vaccine or a control, a placebo. So it's exactly the same way of delivering something. It is actually a vaccine targeting something else. And to give thousands and thousands and thousands of people these vaccines and ask them to go out into the community and basically assess whether or not they got infected by the virus. Okay. And they reported that in different ways, by either symptoms or testing and what have you. But the nutshell is that people that got those vaccines were pretty much not infected, and people that got the placebos were. And we know this to incredibly powerful statistical significance, because usually a phase 3 trial has a maximum of about 10,000 people enlisted for any drug that's ever been developed across the planet in all of history. These trials, every single trial that led to a vaccine being approved by the regulators, had upwards of 40 or 50,000 people recruited. Okay. And that's huge. That gives you such huge confidence that these things work. It also allows you to spot potential side effects that you might not spot in a smaller trial, although we'll come on to side effects in a moment. But these were incredibly powerful trials, and they haven't just done one. I mean, one common thing that people sometimes say is that these things are still in trial. Yes, there are more trials going on because they're testing them in different sorts of patients under different circumstances and in different conditions and different combinations. But the original trials that led to the regulators approving them were done and dusted by sort of autumn last year. And there was huge, rapid scrutiny of those data as well, which led to them being approved. Now, they were approved under an emergency regulatory authority, but that's literally, again, to speed up that red tape process, and they will be ratified in time. That I assure you of. In fact, I think some of them have been. So the doubt that many people have about these untested medicines is fair enough, but it's just important to understand that it wasn't any kind of shortcut that was taken to get those vaccines approved. Okay, so that's really important because they are being used in incredibly huge numbers of people. And in fact, the best validation that we have of these vaccines is what we term phase 4. And phase 4 is the constant monitoring that these vaccines have as a result of being used clinically. So we've administered something like nearly 2 billion doses of vaccine across the planet now, okay? Disproportionately in rich countries, which is a different issue that we can talk about another time. But we've given these vaccines to a huge number of people. And that is tremendously powerful, because we're starting to see the vaccines breaking a link between infection and severe disease. We're starting to see it's not gone, but it's there. And the more people we vaccinate, the better. And there are two reasons for that. One is because, of course, people that are very vulnerable to this virus and can potentially get very sick because perhaps their immune system doesn't work. They may have cancer, they may have autoimmune conditions which require medications that suppress their immune systems, or they might just genetically not respond to vaccines very well, or infection. If you can give them these protective vaccines, that's immensely powerful, because you can safeguard people that you know are vulnerable. And we started doing that with our vaccine rollout. But the second aspect of vaccination is literally safety in numbers, because the act of having a vaccine is actually mainly altruistic. And the reason for that is, if you remember, I said about the virus being an obligate parasite, it needs us to survive. Well, if we essentially do a bit of a Priti Patel and render ourselves as a hostile environment, or probably Theresa May actually originally, wasn't it? Then we limit the opportunities for that virus to thrive and spread. And if we can make sure that the vast majority of us, so somewhere near 80, 90% of us, are a hostile environment for that virus, because we've all got our memory responses ready to go, then that virus has nowhere to go. And because it only has a certain ability to infect more people, the chances of it passing from one person to more than one person, and so increasing the infection in that community, diminishes. And that's what we term this R value that you may have heard, Chris Whitty and Patrick Vallance talking about on the briefings. This R value is a measure of the ability of that virus to either pass on to people if it's above 1, or to only pass on to people some of the time, in which case your epidemic starts to shrink. And that's really important because we know that our restrictions can help with that R value being below 1. We know that vaccines can help with it being below 1. And we know that surveillance and isolating and all those sorts of things help with that too. And so the eventual goal of vaccination is to achieve herd immunity. Well, actually, herd immunity has been tainted, so I'm going to call it population immunity. Okay, so if enough of our population is covered by a vaccine, then it gives that virus nowhere to go. And you can start to see this happening in our country and elsewhere where vaccine coverage is increasing. We're not there yet, and we have a long way to go. And unfortunately, well, actually not fortunately, I have no issue with this at all. It will require adolescents at least to be vaccinated in order to achieve that level of coverage. Okay. You should not do it by natural infection, because if you remember a brief mention I made of the virus being able to sort of evade our immune responses partially before, we know that the responses we make to natural infection aren't as strong or as long lasting as that which we see from vaccines. Okay, so that's the whole idea of vaccinations. That's how they work. That's why they're safe. That's why they're effective. Now, I think the thing to do now is to move on to a few of the really key issues that people have concerns about. But again, if anyone has any questions, then oh, sorry,
James, you had your hand up, didn't you? Did you want to ask a question?
Sorry, I can't see everyone on the screen, I don't think.
I do. Hello, I'm James Allen. Well, the question I had was, are you saying that the foundation of the development of the vaccine is built on years of experience?
Yeah.
And actually, the recipe that you referred to is the bit that's been kind of rushed, has been progressed quickly. So actually,
yes.
In reality, the vaccine's been developing for years.
Yeah.
It's just the finessing of it. Is that a different way of putting it?
Yeah, absolutely right. So if you think about the vaccine technology is just a way of delivering that information, right? So I mean, it's quite staggering, really. This virus emerged in China at the latter end of 2019. We had sequence data available in sort of mid-January, to the world, that is. And we've gone from that to knowing the entire genetic sequence and how this thing has evolved across the planet in staggering detail. So literally, we were able to take that piece of genetic information and transplant it into the systems that were already established in order to program our immune systems in the right way, because it has to be that exact copy of the virus that we use.
I think that's an important point that probably gets lost in all of this.
Yeah, it is. And I see a lot of stuff on Twitter about the vaccines are untested and the vaccines aren't this, that, and the other, and they've been rushed through. And that's exactly why I've addressed that, because it's really important to understand that this is built. Otherwise, they couldn't have done it so quickly, right? You can't go from scratch and develop a delivery system as well as the actual, nuanced piece of information that you want to deliver. So that really would have taken a long time had we started from scratch. And it's actually quite telling that some of the sort of more traditional vaccines, so like killed viruses, for example, aren't working as well. So they don't give quite as good a response as these new technologies. And that's really encouraging, because these really are a sensible way of doing things. Does that make sense?
Yeah, just to me.
Yeah. Okay. There was a question in the chat, Claire?
I think that was
Or was that yours, James?
That was me again, sorry. Yes.
Oh, okay. No, that's great. That's great. Okay.
Steve, just a quick one, sorry, and you might cover this later, but obviously there's now talk about a booster. So if we've had the double dose of our vaccination, is the booster because there's a different variant and a different strain? Can you just perhaps touch on that?
Sure. So there are ethical reasons around boosting, which I won't go into in terms of vaccine equality and stuff. But the reasons for boosting are that people who are clinically extremely vulnerable often they're either quite old, or they may have problems with their immune responses, which could mean that on that spectrum of response that we make to vaccines, their response is suboptimal. Okay, so sometimes that means that it's not quite the right sort of response. Sometimes it means that that response doesn't last as long. And another important thing is that our antibodies tend to peak and then wane, but then plateau. Okay, and most of us will plateau at a very good level, but some people won't. So the rationale for having a booster is that it gives you another kick. You have another capacity to make that better response. So it really does boost things up even higher than you otherwise would do. The other reason for it is variance, as we say. Now, the key thing is that our vaccines still work against all the variants that we know in terms of preventing severe disease. There is some evidence, and there is good evidence, that things like the Delta variant, which is our issue at the moment, can sometimes break through the protection that our vaccines afford at the moment, especially if you're in one of those more vulnerable groups. Okay, that's why it's important that we have two boosts, because two injections, because one injection just gives you that initial response, which is weaker. The second one gives you that higher one. And so to boost that further is, generally speaking, a good idea. Now, there are different ways of going about this. We can either keep boosting our vulnerable people in the way that we do with influenza, or we can strive to achieve that population immunity, in which case, if you've got no cases of virus, you don't need to worry about boosting people. But this argument feels a little bit lost on the government at the moment. Anyway, so the idea is then that not only can you boost and tweak your vaccine to make it better against these variants, because remember, these were made right at the beginning of 2020, so they actually encode the sequence from the original Wuhan strain. Okay, it's been tweaked so it's better at making an immune response, but it's still the original Chinese strain. And this virus is changing because it wants to infect us and transmit better. Okay, it is now also starting to experience our immune responses, and so the more successful viruses start to thrive that can partially avoid those. But there's a kind of parabolic relationship between getting this done. So if you've got a low level of vaccine coverage, and lots of virus, that's a bad idea, because you'll start to make that virus have a race with your vaccine responses. You need to get your vaccine coverage right the way up, in which case you quash that virus down and it can't evolve to become an escape variant. But the danger with the strategy that we're seeing at the moment is that we're going to have very high levels of viral replication and suboptimal vaccination coverage. So I'm personally not in favor of what the Prime Minister announced yesterday, but that's a virological argument. Okay. So shall I move on to more concerns, Claire? Does anyone else have any other questions? So one big thing that people worry about is fertility. Okay. There's been a lot about fertility, and that the vaccines will somehow prevent children being conceived, or could spontaneously cause fetuses to be aborted. I'd like to say from the outset, this is dangerous, poisonous nonsense that was spread by a certain individual in an attempt to try and bring a lawsuit against the European Medicines Agency to get the vaccine stopped. I don't understand why he's done it, but this is what he did, and I'll explain why. And because we've talked about how our immune systems work, we can understand why. Okay. So this guy is called Michael Yeadon. He sets himself up as some kind of fringe hero about scientific wizardry. He talks absolute crap. And the reasons for this are twofold, okay? So what he said was that part of the sequence, part of the recipe that is encoded by the vaccine was the same as a protein that is made in placentas, therefore you would be priming your immune system to attack the placenta when it forms. Okay. So, I mean, I have a real problem with this, because, talking about something like this, which is blatant disinformation about something that's so emotive and important, is criminal in my view. Okay. And the reasons that Michael Yeadon's talking nonsense are twofold. One is, if you remember, at the beginning, our immune responses are trained to get rid of all those soldiers that will recognize our own proteins. Okay, right? So we get rid of all of those ones that could possibly have a problem. And there are some nuances there, people with autoimmune conditions, for example. Okay, But the second problem with what Michael Yeadon said, I'm afraid, is that he showed that the similarity between the vaccine and the placental protein, which was something called alpha-syncytin, which is actually encoded by a virus, by the way, but never mind, was something like 4 or 5 amino acids. So 4 or 5 building blocks of our proteins, okay, a string of that. Now, our immune responses only recognize 8 or 9 or possibly up to 12 chains of amino acids, okay? And that is absolutely programmed into the way our immune responses work. And the reason for that is that if you remember, our genetic code is only programmed with 4 bases, okay? So the amount of combinations that you have there is limited. So actually, if you took a 4 or a 5 amino acid stretch and compared that to a huge number of proteins in our bodies, you would see that stretch in dozens of them. And for example, there's the same overlap that Michael Yeadon noted in a protein called actin, which actually just makes the skeletons of all of our cells. So by Michael Yeadon's logic, we'd all be eliciting an immune response to our cells that would turn us into jelly. And that hasn't happened, right? So it's absolutely scientifically flawed what Michael Yeadon said, okay? And it has troubled and panicked a lot of people. And one of the most common reasons people give, and especially young women, saying that they don't want to have the vaccine because they're worried they won't be able to conceive later on in life. And that is frankly nonsense. It is on an Andrew Wakefield scale of criminal misinformation, in my view. And I hope one day he has to answer for it. So that's my view on Michael Yeadon's fertility issue, okay? There is also stuff about people that have been vaccinated inducing menstrual cycles in women that are in the same room. There's stuff like exacerbating periods and things. Now, the period thing is being looked at. If you're interested, there's a very wonderful scientist called Vicky Mail down in London, and she's doing loads of studies on fertility. I suggest you look at her work, but she's debunking all of this stuff at the moment. Another thing that people say is, well, the vaccines weren't tested on pregnant ladies. And it's like, if you're going to run a clinical trial, you don't start with a possible situation that could go wrong, okay? So you start with normal, healthy volunteers. You don't start with pregnant women. You do your safety trials first, and then you extend it out to pregnant women when it's safe to do so, okay? So all of this about fertility, I'm afraid, as far as I understand, is complete nonsense. So please don't worry about this. And similarly, people talk about long-term effects of the vaccine. Well, there has never been an adverse reaction beyond sort of 8 weeks in a vaccine trial that I'm aware of. Okay, so we would have seen them by now. Okay, but that does lead me on to the idea of side effects, which is another common issue that people have. So we've heard about things like this, these adverse clotting reactions that people can have. There's also this story around myocarditis. Okay. Now, the first thing to say is that the frequency of these things happening is so small that if we were to design a clinical trial to try and assess these things, to have captured it, we'd have had to have tens of millions of people in those trials in order to do so, because they are so infrequent and so rare. Now, that's not to say that they're not a concern, okay? It's absolutely right to say that they are a concern, but any risk that you have is a factor of the chances of it happening and the severity of it. Now, the clots in particular are severe, but the chances of them happening are so small. And especially now, young women under 30 are not offered the AstraZeneca or the adenovirus vectored vaccines anymore, at least in this country. So you don't get that reaction with the RNA vaccines. So that argument for me is pretty much out of the window in terms of the safety of the vaccines, I'm afraid. Okay. There's also this story of myocarditis associated with the RNA vaccines. Okay. Now, that link hasn't actually been formally proven yet. Okay. It's been seen in Israel amongst a handful of young teenage boys primarily. It, again, is a very, 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 very small number of people who develop myocarditis that's actually extremely mild and that actually they recover from without having to undergo any treatment other than a few nights in hospital. Okay. So yes, you will sometimes get adverse inflammatory reactions caused by vaccines. People will get rashes, people will get swelling, people will get fevers, people will get headaches. I personally felt like I had a pretty nasty cold, the second jab of the Pfizer I had in my shoulder, for about 24 hours, and then it went away again. But none of these are in any way as risky or as dangerous as contracting SARS coronavirus 2. And there have been a lot of statistics bandied around about this virus and people saying that there's this huge chance of surviving and all the rest of it. All of these things are percentages, and we're talking about millions of people. So that's why this pandemic is a problem, okay? It's absolutely true that the younger you are, the less likely you are to have a problem, but it's not absolutely true. And as we're seeing at the moment, because the epidemic has shifted into younger people, the people being hospitalized are young people and children, and it's spreading through young people and children, okay? So it's not the case that it's inconsiderable risk to have this virus. It's absolutely unethical, in my view, to allow, for example, as Robert Dingwall suggested the other day, for this virus to pass through our children and young adults without vaccinating them, because that is dangerous. And there's also this aspect of long COVID, which is this post-infectious immune condition, which no one really understands, which does happen for other virus diseases, but in no way to the same degree or frequency. So this virus, if you're talking about the risks associated with your vaccine side effects, which are by and large extremely mild, even if you do feel like you've got flu for 24 hours, on the grand scheme of things, compared with being put on a ventilator in ICU, which is, you know, a small but not inconsiderable possibility, then for me, that balance is a no-brainer. And yes, there are lots of risk assessments going on with different age match groups and all the rest of it. But as well, if we want to try and achieve population immunity, the balance of risk comparing side effects with beneficial effects, which is the same for any medicine that we ever prescribe, any drug, any vaccine, the balance is vastly in favor. And there are numbers to back me up here, which I won't bore you with. But you know, the safety and efficacy of these vaccines is tremendous. There has not been a vaccine against an infectious disease, with the possible exception of the measles vaccine, which, because measles doesn't change very much. These are incredible. They're much better than flu vaccines, for example, and we use those very effectively. So we have the tools in our arsenal, if we're patient, to really see this thing out, but we're being slightly impatient at the moment. So that's side effects. Does anyone have any questions about side effects? There are others, but really, honestly, you know, you will, as I said, we're genetically diverse. We won't all have the same response to a vaccine, but there's a massive bell-shaped curve that we're talking about here. By and large, the vast majority of us will make a good response and a safe response to a vaccine. There will be tail ends either way that either don't respond or that have an unfortunate over-response and an overreaction to it. That is just the nature of medicines. But thankfully, that tail end for these vaccines is very, very small indeed. Okay. So another common thing is that people say they've already had COVID. Why do I need a vaccine? Okay. Well, as I said before, the response that we get from a natural course of first infection with this virus is suboptimal. Okay. And we see this in the seasonal coronaviruses that causes coughs and colds, actually. We get reinfected with those every few years because this virus has a way of circumventing some of those processes. So the traps and mines that we lay in our innate immune system, and actually in some of the aspects of the adaptive one, which means that our responses are not brilliant, okay? So, but the good thing is that you've got that first response there. So if you then have a vaccine afterwards, you get a huge response after you're vaccinated, and another vaccine just tops that up. So it's all good. It's all good. So really, absolutely, if you've had COVID, it really doesn't mean that you shouldn't have your vaccine. And again, this is why Dingwall was really talking rubbish. And I'm sure some of you have heard of the Great Barrington Declaration, which has been widely debunked by the vast majority of scientists, and yet is rearing its ugly head again at the moment. Please, please, please don't consider huge swathes of natural infection to be in any way ethical or safe or actually effective in terms of generating population immunity, because it will not be. So there we go. There's a couple of questions in the chat. Hold on, sorry. That's from James again. Is it true that vaccine reduces symptoms that may arise from variants? Yeah, so the variants of concern are all protected by vaccines, by and large, in terms, it depends on the measure you're talking about. So as I said before, I think they all protect against severe disease and death. That much is clear, very high percentage. And remember that the numbers that people use when they say 95% effective, that doesn't mean that 95% of people will get it and will not get it and 5% will. What they're talking about is a 95% relative risk reduction of you having it. Okay, so if you had a 10% chance of getting COVID, your actual chance after you've had your vaccine would actually only be 0.5%. Yeah, does that make sense? Okay, so they absolutely do protect by and large against severe disease and death. They do protect by and large against most variants against actually being infected, not always and not very well when you've only had one dose. A balance.
Oh, we lost Steve.
Change so that some of the better antibodies don't work as well, but you still got other ones there as a backup. Okay, so that's why it's important to make sure everyone gets 2 doses of those vaccines. There is a danger that the virus could change one day to completely avoid the best ones, but it really is a long way off, but we're not doing us any favors at the moment, unfortunately. And again, if you do have the unfortunate experience of being infected once you've been vaccinated, the chances of you having a severe course of disease are very, very small. It can happen, but it's very small. And also the amount of virus that literally exists in you is reduced dramatically. So you're very unlikely to then pass that infection on. So again, we're going to be pushing that R value below 1 because you're not going to be as infectious as anyone else that hasn't had a vaccine that gets infected. Sorry, there's another question. Why or how does the risk change in relation to ethnicity? Okay, so there's several things happening here. So the risk of severe disease in certain ethnic groups when you're infected by this virus is actually heightened. Okay, and that's linked to some genetic differences, racial differences in some of those innate responses that we make. So the minefields and the tripwires and that sort of thing, we seem to be less good at kicking that off in certain circumstances. And there have been some genetic studies that are starting to understand that, but we don't really know why. But it is important because studies have adjusted for things like occupation, demographics, and all the rest of it, and still found that people from certain ethnic backgrounds still have a sort of 2 to 3 enhanced chance of developing severe disease. And the flip side of that is that it's really unfortunate that due to various socioeconomic reasons, people from those backgrounds are also less likely to take the vaccine up. So there's a huge effort going on at the moment to try and push the message that having a vaccine is really important. So it really is important that we look at those, and also in younger people as well. We're seeing the take up of the vaccine start to drop off, and I'm absolutely certain that that is at least somewhat partly linked to this idea that we can just take all the locks off the wheels and everything now and just go crazy. But there is a real need, especially in those sorts of populations, to get the vaccine out there because they're more at risk, but they're also less likely to take it up. So that's a real job to do as we go forward. So I've discussed, there was another list here about stopping spread, but I think I just talked about that. So the vaccines do prevent you being infected to a degree. They also prevent it passing on. Not perfectly, but pretty damn well. Will I still be wearing a mask? Yes. Yes. Even though I'm double vaccinated, even though I am, relatively youngish and fit-ish. I don't have any underlying health conditions. I will, because I'm aware as a virologist that there is a small possibility that I could be infected and therefore infectious. I also am married to a clinically extremely vulnerable wife, and she has had her vaccines, but all the more reason why, just taking this very simple precaution, which again is actually altruistic mainly, much like vaccines, because masks, the biggest job masks do is limit the amount of virus that you pass on from your breath. They do protect to a degree, and the better fitting mask and the better protection mask that you have, the more protection you get yourself personally. But what they do is they prevent the droplets and aerosols that are coming out of your breath that are infectious where the virus hangs out. So yes, I absolutely will be wearing a mask, especially in crowded indoor spaces, which I will probably avoid anyway, because the estimates are that cases are going to escalate, as we've seen as a result of the football, I'm afraid. Is there another one? Yeah. So we've talked about vaccination doesn't stop spread. Why bother? Yes, it does. I've heard the side effects can be significant, at times worse than COVID itself. I think we've covered that as well. Is that okay? The vaccine was produced really quick. Is it safe? Yes, we've done that. Oh, there's one. Sorry, the vaccine changes your DNA. Is that one that we would like to cover? Okay. Now, the reason I told you about the central dogma, that DNA makes RNA, makes protein, is that that's exactly why the vaccines do not change your DNA, because they make RNA. Okay. RNA does not go back into DNA unless you are a special kind of virus called a retrovirus, which HIV is. And that encodes a special enzyme that's actually able to what we call reverse transcribe the RNA information into DNA. Now, there was a paper out, and I won't shy away from this, there was a paper out in PNAS recently that showed that certain fragments of that virus can be incorporated into the DNA of cells in a culture dish. And there are various flaws in that study, which I won't go into because it is getting into geekdom, I'm afraid. But the reasons are that basically centered at the fact that they use cancer cell lines, which we all use in the lab primarily, and their DNA is inherently unstable, and it breaks apart and comes back together again. And there are enzymes that could make short strands of DNA, and that's probably why they saw something in there, but it's probably actually an artifact of the way they looked. So that paper, I'm afraid, is not widely accepted. But honestly, no RNA virus, hepatitis C, polio, cold viruses, coronavirus, they cannot integrate into your DNA, okay? And even if they did, that cell, the one cell that it happened in a 20 billion would be killed by our immune response, and nothing bad would happen anyway, okay? So I think that's as much as I've got on my list. If anyone else has anything that they would like to ask, then please do feel free. And if I've not explained something properly, or if you feel I've upset anybody, then I do apologize. But yeah, I really do hope that that's covered most angles. Claire, have I missed anything?
I don't think so, Steve. It's been fantastic. Any questions from anybody?
Yeah, I just have a quick one. So assuming that antibody tests are starting to become more available to people, how would an antibody test pick up the difference between if someone's been vaccinated and if someone's actually had the virus? How does that differ if the proteins are trying to replicate the virus?
Okay, so there are several types of antibody tests. There's the sort of lateral flow type tests, which we're used to for actually testing for antigens for the virus itself, sticking the thing up your nose. But obviously you'd put your blood on this one, okay? Those are qualitative, okay? So they tell you what sort of antibodies you have, all right? And the difference between a natural infection and your vaccination is that all your vaccines, at least the ones that are used in this country, only have the spike protein there. So you will only have antibodies to the spike protein. If you've been infected, you will have made antibodies to other parts of the virus. So the main one that they look for is the N protein, which is that protein shell that protects the viral genomic material. Okay, so lots of the lateral flows that you get have got 2 bars on the test thing as well as the control, and you'll have an S bar and an N bar. And so if you only have an S reactive bar, then you've got vaccine protection. If you've got S and N lighting up, then you've been infected at some point. The exception to that is if you've had one of the killed vaccines, so the Sinovac, for example, which is a killed virus vaccine, then you would probably mount a response to N as well. Okay. But in terms of understanding how strong your response is, that has to be done in a lab, I'm afraid, because you have to do a quantitative assessment of the titers of antibodies that you have, and ideally test your serum's ability to stop the virus infecting cells in the laboratory. So you would titrate the antibodies from your blood into a cell culture dish in the presence of the virus and ask, does that stop the virus infecting those cells? But you have to be trained at biological containment level 3+ actually, and work in a glove box and dress up like a Teletubby and work in a negative pressure room, which is the sort of thing I enjoy, which I don't expect many other people to. So yeah, I mean, but that is it. So yeah, the problem is with antibody tests is that they're slightly unwieldy, because obviously you've got to go around stabbing your finger and stuff. So you can get them. In fact, the most common ones that people have are that they will send you a tube which you put some blood into, and you send those off, and they'll assess them for you. Most of the commercial ones are actually pretty good, but the ones that Public Health England recommend are the Roche and another one which I can't remember. But yeah, they're anything that's been accredited by PHE would be worth looking at. But in terms of rolling them out, sort of mass testing is a bit difficult, and there are problems with mass testing anyway, because you need to really target it, although mass testing may become relevant pretty soon with the case numbers we're likely to see.
Thank you.
Is there another one in the chat?
Anything else? Anyone else?
Hold on, I've got this here. A staff member is fearful as they have heard that an ingredient in the vaccine is from animals. Is this correct? The only element that I know of that could possibly go into the vaccine is that this isn't for the RNAs, but for the adenoviral vaccines, the cells that they grow those in, because you have to literally, so that the cells replace the part of the adenovirus that's missing, so they can grow on those cells, and you can make lots of virus, which then if you take it away from the cells is safe. Okay, so remember, it's just a delivery, it's like a carrier pigeon. Okay, those are grown usually with serum from animals, so from cows. Okay, but the virus particles are purified away from all of that, so that there will be no contamination, and they've done lots of validation on that. There is no contamination whatsoever of serum, because they wouldn't be able to use it otherwise, because it has to be clinically approved, it has to be made something called good clinical practice standards. So there's no way that there's any contamination coming through those.
I think what their concern was, Steve, is people who are vegan or vegetarian. It's got animal bits in. I don't want that.
Yeah, well, it's not in the actual vaccine, but I would say to them, get the RNA one, because that's not at all. Yeah. Oh, here we go. With infection increasing in younger adults, what are your thoughts on ending track and trace or isolation when in contact with a positive case? I think it's daft. Frankly, to stop any epidemic, you need to understand where it is. I mean, we mistakenly stopped testing back last spring when we knew that there was imported cases from Europe and elsewhere, and for that reason we had this enormous wave in spring. You need, as I said, the way this virus spread is because it needs us, right? It needs us as a host. So if you carry on, regardless of whether you've had close contact with someone who's infected or not, then I'm afraid you're carrying a risk of that infection not going below R=1, and you're carrying that on. So I don't understand the logic. I think the logic is that they are expecting so many cases with unlocking that they're not going to be able to keep track. Now, one thing I would say is that the testing capacity and the genetic epidemiology in this country is fantastic. fantastic. What is not fantastic is the contact tracing element, because it's this privately run, centralized piece of crap, frankly, that Serco and Cytel have set up, and that doesn't work. I mean, if you compare it to the NHS test and trace, which is, if you remember when they used to separate it into tier 1, tier 2, tier 3, not tiers, what was it? Pillar 1, pillar 2, pillar 3. The effectiveness of the pillar 1 contact tracing was something over 95%. Pillar 2, not so good. So for every contact you're missing, and, you know, with the app pinging off, you know, I agree with Keir Starmer's analogy of taking the batteries out of the smoke alarm. Actually, it's you can't just put your fingers in your ears and start whistling. This is going to be there, and it's a sad fact that we have this link between the vaccine, between infection and severe disease, very much weakened, and that's wonderful to see. But unfortunately, it's not broken. And you can see that now there is an increase in hospitalizations, and deaths are starting to pick up. And they are, unfortunately, not only in younger people, something near 10% of hospitalizations are in children, but also the virus is now finding, and this is another reason for boosters, actually, you need to do both. You need to do population immunity and boosters, that because the virus is so prevalent, it's now finding those people in the population that may not have responded well or have not had their vaccines. So you're starting to see over 65s creeping into hospital again. So this is the problem with mass infection, is that it's going to find people. And for me, I therefore think that what they're doing at the moment, and this is my personal view, is irresponsible and is a reckless experiment. And the reason we've had such a tough time is because they've not made good on 3 lockdowns, so they obviously don't have the courage to do another one.
Steve, you mentioned about children there and the effects of, sort of how they're catching it. When do you think they'll start pushing the vaccines through to children, school-aged children?
Well, that is something JCVI are going to comment on soon, I hear. It's been approved. So the regulators have approved it in 12 and above. In other countries, they've approved it. And in clinically vulnerable children that are younger, they can have it too. All the trials were done not quite to the same scale as the adult trials, just because literally it's harder to recruit kids to trials, but they were all very safe and incredibly effective. There were virtually no infections whatsoever. So I think it's Pfizer that's approved for 12 and above at the moment. I think the AZ are ongoing, but I think that Pfizer is more likely to be the one, and Moderna, that are given to children. And if you look at where the infection is at the moment, it's in basically 10 to 29-year-olds. So that is a real problem, because it is spreading through schools. I find it quite frustrating that people have been sort of gaslighting us in the government, saying that there's no issue with school transmission. And then yesterday, one of the big justifications for opening up now is that it's summer holidays, and so it won't spread through schools. So you work that one out. So the spread through young people is a real risk. Of course, they're less likely to get severely unwell, but they are still potentially able to get severely unwell. And there are children that die of this disease, unfortunately, only a handful, thankfully, but we haven't seen the sort of case prevalence that we're about to see. And of course, there's long COVID, and long COVID in children happens to similar levels as happens in adults. And there are charities all over the country now springing up, supporting families with kids. I mean, if you think about the impact on a young child's life of being ill for a year, I mean, that for me is just, I mean, my young boy was ill when he was very young. He had meningitis and some other problems after birth. And that was one of the most traumatic periods of my life. And to have a kid that's ill for that long, it just must be dreadful. So, I'm not trying to be emotional about this. If we are going to achieve population immunity, I'm afraid the numbers, they're given the percentage of adults that have got their vaccine. That doesn't matter, because the virus doesn't frankly care how old you are. It will infect you nonetheless. And that is the problem that we have. It's very clear that they are willing to take this hit through natural infection, and they are mistaken if they think it's going to reach population immunity that way, in my view. Sorry, there's one about anaphylaxis here. Anaphylaxis, still not advised to receive the vaccine. That's a specific a reason not to have the Pfizer vaccine specifically, I believe. I don't think it applies to Moderna, but certainly it's okay to have the AZ, I believe. So, but the anaphylaxis, is only people that are certified, having to carry EpiPens with a clinically diagnosed risk of anaphylaxis and allergies. So yeah, I mean, that is an unfortunate aspect that would apply to many vaccines, I'm afraid, but this isn't in particular an issue with the Pfizer, I believe. But this is another great thing. We've got so many different opportunities now. We're about to have the Novavax vaccine, which is actually it's a protein subunit already made. So there's no need for any genetic material whatsoever. It's made in insect cells, and you can have that a little bit like the hepatitis B vaccine. It's just protein. So that could be a much safer option for people that might have this sort of anaphylactic response. So actually,
any more questions? No? Okay, so for everyone on the phone and on the call, thank you for joining us today. If you or any of your teams would like any kind of follow-up one-to-one basis, if you could let me know, claire.leake@nationalcaregroup.com, and I can get that organised. But I hope you found it useful.